Hi, my name is Rauf. I'm a research fellow at NCI NIH. This research is regarding genetic regulation of OS1 and how that affects COVID-19 outcomes. Response to pathogens, including SARS-CoV-2, is likely determined by the interaction between host and pathogenic factors. GWAS have reported a series of genetic variants in this regard. One of the genetic variants is on chromosome 12 near OS1 to 3 gene cluster. These are interferon-stimulated genes that encode antiviral proteins. These proteins activate RNase L pathway that degrades the target double-strand RNA, which in this case, viral RNA. We use case analysis, functional genomic approaches, and data from clinical trial with recombinant interferon lambda to further understand the mechanism of this genetic locus with COVID-19. In COWNET study, we found association of genetic variants in OS1 with increased hospitalization and increased risk of developing severe COVID-19. The genetic variants of OS1, their representative haplotypes further predicted this association. One of the genetic variants in OS1 generates two major isoforms, P42 with risk allele and P46 with non-risk allele. By generating plasmids that can represent some of the haplotypes and testing their anti-SARS-CoV-2 activity, we found that all isoforms had similar anti-SARS-CoV-2 activity. To further understand the regulation of OS1, we evaluated RNA-seq samples. We found that in heterozygous samples, the expression of transcripts was higher with G allele compared to A allele, representing allelic imbalance. With further analysis, we found that allelic imbalance was mainly driven by genetic variant in exon 3. Using mini-gene assays and evaluating splicing pattern of exon 3, we found it had two splice isoforms, long and short. Long isoform is highly expressed in the presence of G allele. With further analysis, we found that knockdown of nonsense-mediated decay leads to upregulation of long as well as short P42 compared to long P46. P42 is present in the presence of risk allele, suggesting that loss of expression in the presence of risk allele, particularly P42, is due to nonsense-mediated decay. To conclude, in Europeans, the increased risk of hospitalization is associated with genetic variants of OS1, and regulation of OS1 is mainly driven by two genetic variants that contribute to impaired anti-SARS-CoV-2 activity and elevated risk of hospitalization. Data from clinical trials suggested that interferons can overcome genetically driven response to SARS-CoV-2. This is a collaborative effort of our team with several teams all over the world. The manuscript is available as preprint in medRxiv. I'll be happy to answer any questions. Thank you.